we have the pleasure now to have Professor Samraat Pawar. Samraat obtained his PhD degree in theoretical ecology from the University of Texas at Austin in 2009. After that, he was a postdoc at UCLA and University of Chicago and joined the faculty in the Department of Life Science, Imperial College London in 2013. His expertise lies in integrating mathematical modeling, computing, and empirical data from biology, chemistry, physics, and complex dynamic systems. He's particularly interested in how environmental temperature and biophysical scaling laws together mediate the emergence of stable ecosystems, including microbial communities. Samraat's most recent work has been focused on using information about cellular level metabolic and biophysical constraints to predict and engineer the formation of stable metabolic and biochemical interaction networks with complex microbiomes. It is a pleasure to have you here and looking forward for your presentation, Samraat. Thanks.
So I just wanted to talk today about some new work we are starting. It's led what I'm talking about today. 10 years of work has gone in leading to this, and I'll hopefully give that context as I go through the talk. So my title is a bit more radical than the original one was. It is about how it's basically is engineering microbiomes for the real world. And if you have any questions about what this logo or whatever you want to call it really means, happy to clarify that later, but let's leave it at that. So I'm going to talk about microbiome engineering in a broad sense and try to define it in a broad sense and talk about why temperature is such an important factor to think about, and then talk about why how we can actually incorporate the effects of temperature fluctuations into microbial community engineering. And then I'll talk about this project that I just mentioned that has just started. So I know this audience probably is familiar with this, but I thought I'll define it broadly, to get everybody on the same page, at least from my perspective. Microbiomes, what are they? There is some discussion out there, but generally it's the microbial community combined with the collective genetic material and the surrounding environmental conditions, including metabolites, nutrients, and I guess changes in the thermal environment also, but generally driven by the microbiome itself. Just to give you an example of a real-world microbiome that people might might be interested in, or might has got important functions, is the termite gut microbiome you can see on the screen. It's basically a metabolic interaction network for lignocellular digestion, which is what keeps termites happy and alive, because they basically live off wood. And this one example, and there's probably an underrepresentation of the true complexity. There are already 205 microbes, strain species, whatever you want to call them, about 265 metabolites. So you can see that these kind of microbiomes, the real world microbiomes, are extremely complex. Microbiome engineering, there are several applications, and Jürgen just gave a really nice talk about one of those examples, and I think my talk hopefully complements that and leads to some discussion. But essentially, we are interested in optimizing, harnessing, and manipulating, and then assembling microbial communities that can achieve specific beneficial functions, like for example, enhancing gut metabolism, in agriculture, promoting plant growth, environmental sustainability, bioremediation of waste treatment, and then industry and biotechnology like biofuels and high-value chemicals, which is the kind of thing Jürgen was talking about earlier. And here below is just a collection of some figures I got off the web, people talking about these kind of applications and potential. And then, of course, the largest scale problem is ecosystems. So we would want to understand how ecosystems can be regulated or restored, and how can microbes contribute to that, and why is microbiome engineering such an important thing at all of these different applications and really all of these different scales, because we're talking about bioreactors that are very localized to large scale ecosystems, which might be talking about hundreds of squares of kilometers or more. Well, the simple fact is, and again, probably I'm preaching to the converted here already, microbes are the unseen majority. And this is one of my favorite papers, and it tells us something that it was only published in 2018. which means that till 2018, there wasn't really a good estimate of this. And even this estimate that you're looking at is still, obviously is subject to a lot of error, but it is about in the ballpark, at least, that we know now of what role microbes play. So the short end of the story basically is that basically plants, bacteria, fungi, archaea, protists, and arthropods dominate Earth's biomass. That's 99.3%. And out of that, bacteria, fungi, and archaea are about 17, and in fact, probably quite a bit more, maybe 20 or more percent of Earth's biomass. And so essentially, among plants also, we're talking about phytoplankton. So these are obviously microbes as well. So essentially, microbes make the world not go around, but make it work. So everybody does want to do microbial community engineering. And I'm just going to, just flip through a whole lot of papers. And if you look at the dates, these are all papers that have been emerging in the last literally 2 to 3 years. Right? So everybody wants to do it. One thing to notice about all these papers are that they are all opinion papers. And we currently are very much at the beginning of this enterprise of how do we engineer or control these very complex systems. And how are the approaches? What are the approaches people are using? So broadly, people are using top-down versus bottom approaches. And I would say, for example, the previous talk, Jürgen's talk, was more on the bottom-up end of things, where you would want to assemble rationally microbial communities from the ground up by putting strains together that have been artificially selected or can be engineered or have synthetic strains, or they can be natural strains, but we are still building them very strategically from bottom up. And the top-down approach is essentially to manipulate whole existing microbiomes and then winnow them down, or attenuate down to communities, sub-communities, or substates, or subspecies compositions that do something beneficial. So basically, there's a couple of figures from a couple of papers. The left figure nicely illustrates this top-down versus bottom-up approach. And they both basically result in what we really want to get at, everybody wants to get at, which is like a predictive mathematical modeling framework or a theory that will allow us to essentially assemble and control these complex systems. So design, assemble, and control. So why am I talking about temperature in all of this? First of all, and I think, again, most people, nobody would disagree with this, but just want to make it clear that temperature is probably the most fundamental driver of microbial physiology. And I say probably, but really it is, because temperature controls enzyme kinetics. It controls the amount of energy that is being consumed in any biochemical reaction or being processed. So essentially, we all know, for example, that the kcat of an enzyme behaves in an Arrhenius way up to a point, and then the enzyme function declines. Also, you can see the same phenomenon at the level of the whole microbial physiology. So what you're seeing here is growth rate versus temperature of a particular cyanobacterium. And what you're seeing here is that it goes up, the temperature performance goes up with up to a point, and then it declines quite radically, quite rapidly compared to the rise. And there are several thermodynamic models that can predict this, but the key point is that this kind of thermal performance curve, if you like, is a very cardinal form of curve. And the growth rate that you're seeing here emerges from the enzyme kinetics, enzyme-controlled biochemical reactions in the order of thousands of kind of enzymes that are controlling. So in E. coli, for example, you'd be dealing with about 2,000 enzymes or more. The papers below are papers from our lab group and collaborators that have been publishing ideas and approaches and results and models about how temperature controls microbial physiology across the tree of life. And what's important from in these kind of performance curves is, one is this activation energy, which is basically the energy barrier that needs to be overcome for an enzyme for a biochemical reaction to move into an autocatalytic phase. And essentially, the lower the activation energy of a particular biochemical pathway, or for the whole organism's metabolism, more or less is the energy barrier to be productive or be fit. And essentially what it means is that organisms or physiologies with low activation energies tend to perform relatively equally across different temperatures, and those that have high activation energies tend to be very sensitive to temperature, so a little change in temperature can rapidly increase the performance. And the T peak, or the thermal, the peak here is basically the temperature at which that enzyme function or organismal fitness is maximum. And I'm not calling it an optimum because there's no guarantee that a enzyme optimum is the growth rate optimum for an organism. And there are several generalities in how temperature affects individual cell physiology and growth. So these are single strain cultures, what you're seeing here, of heterotrophic prokaryotes, so basically bacteria. We do have results for archaea as well. And you see two kinds of phenomena, and this kind of general pattern is really important to think about in biochemical engineering or microbiome engineering, because you might be interested, for example, in engineering microbiomes in what people would call the mesophilic range, which is a very common range in which organisms and bioreactors exist, which is up to 20, 40, 45 degrees. And then you've got the thermophilic range, where you might be dealing with organisms that live in very hot temperatures, but also have very high rates of bioproduction, but they are limited by the ability to adapt. So what you can see from this figure, and this is one of the papers that I've shown before, so you can find this figure there. But essentially, up to the mesophilic range, we know that as bacteria adapt to warmer temperatures, they can increase in fitness. Their overall bioprocessing rates can increase, but then they hit a ceiling, and that's limited, and that ceiling is hit basically through other constraints like the metabolic costs of heat shock proteins, a reduction in enzyme functioning, for example, partial denaturation, or limits on membrane fluidity. Membranes become too fluid and bioproduction is limited. So these kind of general constraints across lots of bacterial taxa are important for us to keep in mind, because when we go down to the engineering problem, we can harness some of this variation, which is what I'm going to talk about. In general, then, we know that hotter is better up to a point within a particular mesophilic temperature range for bacteria. And also, this variation, for example, in activation energy, varies quite systematically. So if you're trying to assemble a microbiome that is a mixture of 2 or 3 different microbial strains, we know also that those microbial strains will be limited in the ability to evolve to a specific temperature performance, because different microbial groups have got very characteristic temperature performance curves. And that's another thing that we need to keep in mind. So here is, for example, a phylogenetically and evolutionarily informed analysis of how the activation energies of whole growth rates vary across essentially most of the microbial tree of life. And you can see that it varies quite a lot. And you can see, for example, the activation energies vary a lot by different types of classifications. And the vertical dotted line is basically the activation energies for eukaryotes. So what we also know is that, for example, fungi have got lower activation energy in general, and they cannot evolve to have the same activation energy as bacteria. And bacteria tend to be more thermally sensitive, but they also tend to be able to live at hotter temperatures. And of course, microbiome engineering is not a new thing. So here's in the middle figure is our daughter many years ago with a failed yogurt culture. But over the evolution of human culture, humans have also cultured bacteria and microbes and fungi, and they have engineered microbiomes. But one characteristic thing about all of these engineered microbiomes is that they work under very specific thermal conditions. And anybody who does any of these kind of things will tell you that. But the real world is not quite like a controlled bioreactor. There are thermal fluctuations. So these are actually ponds at where I work at Selwood Park in England, in south of England. And the left is what the ponds look like. So this is a climate change experiment, and we're assembling basically whole ecosystems in there, including microbiomes. Left is summer, right is winter. And you can imagine that if you wanted to control or regulate these kind of systems, you have to deal with these kind of extreme fluctuations. And of course, then there is a matter of thermal fluctuations or over time that are increasing because of climate warming. Again, something that I don't really need to convince people about, but this is an increasing consideration, especially if you want to do these things at scale, if you want to be controlling and engineering the systems across large scale or across latitudes, in the tropics versus the temperate areas. And then finally, we do have enough circumstantial evidence that temperature is a major driver of microbial diversity and functioning, of course. But for example, here is a paper from Thompson et al. In 2017, where they did a controlled sampling of microbiomes across different gradients. And you can see that temperature and pH are the two biggest control on diversity of microbes. And the bottom left, I'm just citing a paper of ours that partly explains why this happens, and it's using some of the approaches I want to talk about later. So a basic point is that current microbiome engineering approaches rarely work in the real world when we think about temperature. And when I say over the real world in terms of temperature, what I mean is that over time, cultures will collapse because of thermal fluctuations, and over space, because different spatial locations have different thermal profiles, something that's engineered for here might not work there. Why is this? So again, obviously, so Jürgen just talked about genome scale modeling, and he talked about flux balance analysis and constraint made modeling. Current methods largely rely on those methods, and obviously that's going to be fundamentally important when we want to engineer these systems. Because we want to be able to control cellular physiology, understand it, and use it to optimize bioprocessing. But there's a severe limitation with pure GEM and CBM-based approaches, which is that even with dynamic flux balance analysis of the kind that Jürgen was talking about earlier, dynamically changing temperature effects are hard to predict in terms of their downstream effects. So, for example, changes in temperature will change the rate of metabolite bioproduction, which will then result in changes in cross-feeding, which is cooperation or facilitation, and changes in resource competition. So these systems can quickly, the consumer resource dynamic changes rapidly, and you can get unstable systems. So you get culture collapse. And applying control under these fluctuating or controlling the systems at different scales gets really important. So imagine you get into the zone where you want to bake larger bioreactors that do the same thing. It's much harder to control temperature in a larger bioreactor in a nonlinear way than in a smaller bioreactor. So how can we really make this work? This is obviously a hard problem, and it's going to be one of the limitations in our view that are going to prevent bioengineering approaches being really applicable in many scenarios of at the microbiome level, at least. So we've been thinking about this problem for a while, and lots of people out there have been thinking about these kind of problems for a while. And our suggested solution is to use what you'd essentially call the design, test, and build kind of approach, which is a bioengineering paradigm, but apply it in the context of microbiomes by combining three different things. So the Digital Microbiome Project is a software project, essentially, and what we are doing, and we've just started this work, is merge theoretical ecology, and I'm going to give you an example of that real quick in a minute, which is consumer resource dynamics, with the GEM kind of approach. So basically use multi-omics and metabolic network modeling to parameterize the theoretical ecology models, and then use machine learning for dynamic monitoring and control, and reparameterize those theoretical ecology models over time. So we can actually predict how this nonlinear system, where it's going to go, and make changes to the environment to be able to control that. Obviously, this is not a straightforward task. So the concept essentially would be that we would take standard natural variation in microbiomes or engineered variation in microbial strains, and then find the right combinations for different temp thermal regimes. And even with the simplest kind of fluctuation, here I'm showing an oscillation, we would look at temperature in two ways. One is that we would first design different microbiomes that are resilient at different temperatures, and then we would use temperature itself as a way to control the environment. So basically, making it cooler or warmer to keep the microbiome under some relatively controlled state, so that it continues to do what it's supposed to do for as much time as possible. And this kind of idea, in a more general sense, has recently been started to be talked about. So this is Sanchez's lab in Spain, and they recently talked about how optimization of microbial functions. One way to do this is through rational environmental manipulation. And finally, then I just wanted to wrap this up with a kind of a proof of principle of how we could be approaching this problem. So the simplest way we can approach this problem in terms of the consumer resource theory is to use Lotka-Volterra models. And what we have been doing of late is parameterizing the growth and the interaction parameters of these kind of models using the thermal physiology that I talked about. So constraining it using biochemical kinetic laws, but also scaling laws, for example, cell sizes matter, and then kind of coming up and designing or identifying microbial communities that kind of can live feasibly together under different conditions. So here I'm showing you on the right two kinds of feasible systems. The one in the bottom is a much more resource species poor system that probably will not have that many functions, whereas the one on the top is probably functionally more capable, but obviously it's going to be harder to control because it's got a lot more species in it. But so we can look for identifying the right kind of combinations for the right kind of problems. And the red box on the left is basically to illustrate the point that we are focusing on what I would call the mesophilic range. We are not focusing so much on the extreme temperature range yet, because that changes the problem again and makes it even more difficult. But we believe that a lot of the problems right now lie in this mesophilic range. Let's address it there first. And as a proof of principle, my collaborator one of our collaborators in the University of Exeter, Gabriel Jon Durasher and his lab, and Paqui Garcia, they did some experiments. This is the paper at the bottom. But essentially what we did is we derived predictions for what would happen for bioprocessing capabilities of a microbiome, essentially measuring its respiration and inferring its decomposition rates of substrate. And what we predicted is that using this kind of mathematical framework, that when a system switches to cooperation, it will essentially increase the activation energy of the whole system in a nonlinear way. And to test that qualitative prediction, Paqui and et al., they grew 8 single strains pairwise and as single strains as a control. And then we looked at the same species as a community of the 8 species community growing on a very resource rich, like M9 plus glucose media, where they don't really need to compete. They don't really need to cooperate, but they compete for energy rich molecules like glucose. And whereas the same community assembled on spent media where they're forced to cooperate tend to have a higher thermal sensitivity, because what they've done is they've developed positive cooperative interactions, which essentially non-linearly increases the bioprocessing ability of the whole system. So yeah, for example, from shifting from competition to facilitation, even during assembly, we predicted would increase, and we found that sensitivity increases by as much as 60%. So you can get higher respiration by 60%, which obviously is not bioproduction, but you can imagine that this kind of dynamics would be seen in other scenarios if you were to look into it, if you were to start to model them. So ultimately, what I've shown you this here, just as a short proof of principle, is just looking at the very basic idea of, like, in an exponential phase of a community assembly, what would be the nature of the microbiomes functioning. What we need to do now in the future is start to move this forward into fully complex systems like this. And there's several issues to think about there. Like I said, we are just getting started. And obviously, we are very keen on collaborating around this kind of approach and vision, is that we would like to, for example, understand how can we deal with changes in interaction structure in a complex microbiome, changes in interactions at a higher order, like A changes the interactions between B and C. Plasticity, microbes, E. coli can shut down particular pathways. Most microbes can do that, or they can activate new ones. And eventually evolution. So, long-term bioreactors, will they remain stable when the microbes in there are evolving? So how can we control them over a longer period? So to summarize, when I say real world, I mean temperature in particular is a limiting factor in our view of what is going to limit our ability to engineer and apply microbiome engineering solutions. We need to harness the variation that exists in microbial thermal physiologies to address this. And for example, we can quickly reassemble a new community at a new temperature using natural variation. And then I showed you an example of how warming can really amplify when interacting with the kind of interactions in the microbiome, the functioning of a microbiome or its bioprocessing ability. And now we are moving towards this kind of toolkit, which we're just getting started, but is our vision for how we can start to offer up a solution that merges basically consumer resource theory and genome scale modeling and AI-based approaches. Currently, and this is me doing some shameless advertising, we will be advertising for a postdoc next year and a couple of PhD projects. There are two projects where we are applying this. One is for engineering microbiomes of wheat in the rhizosphere, and another one is looking at how we can mitigate carbon dioxide emissions from freshwater systems. Can we introduce microbial communities that can then change the fundamental carbon processing abilities of these natural communities or semi-natural communities? And we're very interested in the first problem. So obviously the kind of talk, the region that Jürgen was giving a talk in is in the zone of industrial bioprocessing. And I did say that that's also a place where I think we need to think about thermal fluctuations. So we'll be very keen on discussing collaborations or getting perspectives from that angle. I certainly want to end with thanking a bunch of people and funding agencies. And like I said, this has been 10 years of work building up to what we think would be an interesting, exciting approach towards addressing this problem of thermal fluctuations. And this work that I showed you has been made possible by all these people spanning theory and experiments.